Hello friends, welcome back to my YouTube channel Nasrin Padasale. So today we are going to study the important topic coming under unit number 7, system physiology animal. Already we studied unit number 9, diversity of life form. Okay, so today we'll cover system physiology animal. Okay, so topic wise I'm going to deliver a lecture friends. So first important topic is with regard to blood group. So blood group generally represents a contribution of two important genes. One is ABO gene and another one is H gene. Okay, so H gene encode an enzyme called glycosyl transferase, and this enzyme is responsible for transferring the monosaccharide to polysaccharide. Next is ABO gene, which is located in chromosomal number 9. Okay, so the structure of blood groups. Okay, so O blood group has a basic oligosaccharide structure. Okay, for different blood groups. Okay, so this O blood group contains two galactose unit, a fucose unit, and a N-acetyl-glucosamine unit. Okay, so with regard to A blood group have a basic. So this is with regard to blood group O friends. So here you can able to find the blood group O. Okay. So the A blood group consists of a basic structure of O, that is O blood group structure. It has an additional unit called N-acetyl-galactosamine unit. So here this violet color is a N-acetyl-galactosamine unit. So please make a note of this particular name. Next, with respect to B blood group, it also have a basic structure of O blood group, but another unit called galactose is present. Okay, so here you can able to see another galactose unit. Okay, so this sandal color is a galactose. Okay, this is a structure of blood group. Next, we are going to study about blood clotting pathway. So it is a pathway of transformation of blood from a liquid form to a gel form. Okay, so blood clot, what is contained in blood clot mean? It is a network of insoluble protein fibers called fibrin. Okay, in this fibrin there are many formed element of blood like RBC, platelets will be trapped. Okay, and the blood clotting pathway consists of many chemical reaction which ultimately form a fibrin threads. Okay, and these clotting factors which are play an important role in blood clotting pathway include the calcium ion. In addition to that, it also consists of several inactive enzymes. In other words, we can say inactive enzyme, another name is zymosin, and these are produced from liver hepatocyte. Okay, and they are released into the bloodstream. Okay, so this is how the conversion of soluble protein called plasma protein called fibrinogen. So fibrinogen is a soluble protein into a insoluble fibrin. Okay, and the process is catalyzed by the enzyme called thrombin. Okay, so to activate this enzyme, another enzyme is playing the role. The enzyme name is prothrombinase, which convert the prothrombin, which is an inactive form of thrombin enzyme, into thrombin. So here you can able to see. So prothrombin is converted into thrombin in the presence of Ca2 plus and prothrombin activator. Next, this thrombin, which is actually a catalysis enzyme which converts this fibrinogen. This fibrinogen is a soluble protein into a fibrinogen monomer. Okay, so this fibrinogen monomer will form many unit and finally form a polymerase substances called fibrin fiber, and this enzyme catalysis is done by calcium ion. Next, this fibrin fiber in the presence of thrombin, that is this thrombin will activate a fibrin stabilize factor. As a result, they form a cross-linked fibrin fibers. Okay. Next, the blood clotting pathway is classified into two types. One is called intrinsic pathway and another one is called extrinsic pathway. In this intrinsic pathway is highly complex and it occurs slowly. Okay. Next is in extrinsic pathway. This is shorter as well as occurs rapidly. So what are the factors that are coming under this intrinsic pathway means factor 1, 2, 9, 10, 11, 12 or include the factors that play an important role in intrinsic factor. Okay. So as I mentioned you earlier, these factors circulate through the bloodstream as a zymosin, and these factors are an enzyme called serine protease group. Okay, so in this factor number 2, 7, 9, 10, 11, 12 are serine proteases, whereas other factors like factor 5, factor 8 and factor number 13 are not serine proteases. Rest of all factors are serine proteases, whereas the factors that is involved in the extrinsic pathway is 1, 2, 7, 10. Okay. Next, this intrinsic pathway is activated by exposing them to a endothelial collagen. So inside the blood vessel there is a endothelium. So this pathway is activated by the collagen, whereas this extrinsic pathway is activated through a tissue factor that is released by endothelial cell. Okay, so after external damage, that is if there is an external damage means this external pathway is activated by a tissue factor, and this tissue factor is released by the endothelial cell. Okay, next, with respect to this intrinsic pathway, the collagen fiber that are present in the connective tissue that are present around the endothelial cell of the blood vessel activates a factor number 12. So starting factor is factor number 12. Then ultimately a series of reaction will occur which ultimately form a factor number 10. Okay, next in this extrinsic pathway there is an important factor called thromboplastin, which is otherwise called as factor number 3, that will leaks from the blood into outside of the cell. So that's how this pathway gets its name, extrinsic of blood, extrinsic to blood vessel. Next, in the presence of calcium, this thromboplastin will activate a sequence of reaction, okay, finally forming a factor number 10. So the negative feedback mechanism. So in order to prevent the overcoagulation which is widespread in thrombosis process, there are certain processes that are being available to check the coagulation cascade in check. Okay, so first this thrombin acts as a procoagulant and it also acts as a negative feedback. How means this thrombin will activate a plasminogen, so that plasminogen will be converted into plasmin and it will stimulate the production of antithrombin, which is otherwise called AT. Okay, so this plasmin directly act on a fibrin mesh and they simply break down the fibrin mesh. Okay, and this antithrombin decreases the production of thrombin from prothrombin. Okay, and it also decreases the amount of activated factor X. Okay, and in addition to that antithrombin, protein C and protein S are also been available in order to prevent coagulation, and both the protein C and protein X they do this action mainly by inactivating the factor number 5 and factor number 8. So these are the factor, blood clotting factor, the source of their presence, from which part of the body they actually originate, and the name of this. So I just took this from a Ganong's book only, so without any confusion you can study. Okay, so in this factor number, all the factor has been originated from liver only, but factor number 3 is originated from damaged tissue and platelets, whereas factor number 5 and factor number 13 are being originated from liver, platelets. Okay, so factor number 4 is originated from diet, bones and platelet. Okay, so in this fibrinogen is called factor number 1 and prothrombin is called factor number 2 and tissue thromboplastin is called as factor number 3. Okay, so calcium ion, another name is factor number 4. Okay, in addition to that, this fibrinase or fibrin stabilizing factor, which I already mentioned in this video, is called otherwise called as factor number 13. So studying this factor is also highly important, friends. So Agamemnon factor, which is also called as anti-hemophilic factor D, is also called as factor number 12. So make sure of reading all the things that I have mentioned in this tablet form. So this is the intrinsic pathway and this is the extrinsic pathway. Intrinsic pathway starts with factor number 12, whereas extrinsic pathway starts with the factor number 7. So this intrinsic pathway begins with the activation of factor number 12, which will becomes a factor number 2A. So after activation this will become 12A. Okay, so this factor called 12A acts as a catalyst in activating the next pathway called next factor called 11. So this is how the coagulation pathway will just move. Okay, so the common pathway result after the activation of factor number 8. So factor number 8 is common to both the pathway. Okay, so this is how the blood clotting pathway will occur. So next is the cardiovascular system. So first important topic is this ECG. So we need to know what are the P wave and what is mean by that and why this P wave has been originated. So first this P wave is upward deflection of ECG, whereas this Q wave is the first downward deflection. Next is R wave is the first upward deflection in a positive way, and S wave is the first downward deflection in a negative way. So this P wave has created due to arterial depolarization. Next this S wave has been originated due to ventricular depolarization, that also late depolarization of ventricle. Next this T wave has been originated due to, that is when ventricle repolarization occur, then T wave will be originating. Okay, so this T wave occur when the ventricle started to relax. Next is the QRS complex. This QRS complex has been originated as a result of ventricle contraction as well as depolarization in ventricle. Okay, so you should definitely know how the waves are being generated. Okay, next is ST segment lies between the QRS segment and T segment. QT interval, it occurs, the time point of QT interval is beginning of QRS complex to the end of T wave, and this ST segment originates as a result of time gap between the depolarization and repolarization in ventricle. Next this QT interval, it is a time between the electrical activity in the ventricle and the return of ventricle to resting state. Next this P wave interval includes the P wave as well as R segment. This P wave is a time taken by the atrioventricular node to conduct the impulse, and this PR interval will be shorter at a higher heart rate, that is due to exercise, and this PR interval will get started to increase due to lower heart rate, that is sleep. Next, so these are the question that has been asked repeatedly. So they are asking S wave of normal human ECG originate due to what, like that they are asking. So you should know S wave originated due to late depolarization in ventricle. So the correct answer is the late depolarization of ventricle valve. So you should know the word late. Okay, so these are the question, you can work it also, friends. So next, in this ECG analysis, the size of each wave provides a clue to abnormalities. Okay, so if the P wave is larger than unusual means, then it suggests the enlargement of RTM. Next, the Q wave is enlarged in manner, it suggests the person is suffering from myocardial injury. Next, if the R wave is enlarged means, it suggests that ventricles are enlarged. Next, if the T wave is flattened than normal, so here you can able to see T wave is already in a flat manner. So if this T wave is flat than normal, then it suggests that heart muscle is receiving an insufficient oxygen, that is due to hypoxia condition. This T wave might be normal. In the other circumstances, if the T wave is being elevated means it includes the blood potassium level are high. Okay, that is hyperkalemia condition. Next, we can see hormonal control of blood pressure. So first hormone is angiotensin 2. So it is a powerful vasoconstrictor, friends. So the mechanism is that when the volume of blood or the blood flow to kidney started to decrease means, the juxtagomerular cells in the kidney will secrete an enzyme called renin into the bloodstream. So this renin is actually a aspartyl protease kind of enzyme, and this renin will catalyze the angiotensinogen. Okay, so this angiotensinogen is actually released from liver. So this angiotensinogen, the presence of renin, will be converted into angiotensin 1. So this angiotensin 1 in turn will be hydrolyzed by an angiotensin converting enzyme in order to form a angiotensin 2. So this angiotensin 2 is actually a octapeptide, and this angiotensin play a vital role in rising the blood pressure. First is acting as a vasoconstrictor. Next is angiotensin 2 enzyme will stimulate the aldosterone. Okay, so this aldosterone will definitely increase the reabsorption of sodium ion by kidney. So ultimately the sodium ion level will be increased. So it will contribute to the retention of water, which will be ultimately lead to an increase in total blood volume. So if the total blood volume increases, then ultimately the blood pressure will be increased. Next enzyme. So this is what, friends. So from kidney, this juxtagomerular apparatus is present. So it will secrete the renin enzyme. So this renin will convert angiotensinogen into angiotensin 1, and this angiotensin will be converted into angiotensin 2 in the presence of angiotensin converting enzyme. So this angiotensin 2 will stimulate the adrenal cortex to produce an enzyme called aldosterone. Okay, so this is what the thing. Next, this angiotensin converting enzyme is actually a dipeptidyl carboxypeptidase. So you should know it is a dipeptidyl carboxypeptidase enzyme, whereas this renin is a aspartyl protease and blood clotting factors are serine protease. Okay, so this angiotensin converting enzyme is a dipeptidyl carboxypeptidase. It actually splits off the histidine and leucine bond. Okay, so that the physiologically inactive angiotensin 1 will be converted into angiotensin 2. So this renin enzyme will split the leucine and valine bond, whereas this angiotensin converting enzyme will split the phenylalanine histidine bond. Okay, so in addition to that, it also splits off the histidine and leucine bond. So the next hormone is epinephrine and non-epinephrine. Both are acting as a vasoconstrictor. Next enzyme is next hormone is vasopressin. It's otherwise called as antidiuretic hormone. This antidiuretic hormone is also called as ADH. It has been produced from hypothalamus, and it has been released from the posterior pituitary in response to dehydration. And another reason of this production of this antidiuretic hormone is that it is also produced in the presence of decreased blood volume. So the main mechanism of antidiuretic hormone is that it will increase the water reabsorption in the collecting duct of nephron. As a result, it will cause vasoconstriction, which ultimately increases the blood pressure. So if the vasoconstriction occurs, it will increase the blood pressure. If vasodilation occurs, it will decrease the blood pressure. Okay, so the fourth hormone name is arterioneuritic peptide. So this is peptide that has been released from the cells of atria, which is present in heart. So this arterioneuritic peptide will generally lower the blood pressure. So vasopressin, epinephrine, and the another hormone name is angiotensin 2 will increase the blood pressure by causing vasoconstriction, whereas this arterioneuritic peptide will lower the blood pressure by causing a vasodilation, as well as it will promote the loss of salt and water in the urine, which reduces the blood pressure. Which in angiotensin, I just said this aldosterone will stimulate the reabsorption of sodium, which will increase the sodium ion. As a result, the water concentration will be increases as a whole. The blood volume will be increased, and the blood pressure will be ultimately increased. So arterioneuritic peptide have an opposite effect of angiotensin 2. Okay, so in the next video, I'll definitely share with you what are the next notes that we are going to study. Thank you, friends. Thanks for watching this video.